Whenever designing the dissolution specifications and designing the parameters and doing the validation, we need to follow all the related guidelines. The guidelines may be from the country specific agencies, ICH, and the product monograph. Then it will be very easy to justify the dissolution specifications. Whenever we make the specifications for some tests, it should have a justification. So this video is regarding the justification for dissolution specification. You might be known to the formulations like IR, ER or DR and some others. Depending on to the formulation, depending on to the country in which you are going to market the product, the dissolution specifications are designed. I welcome all the viewers to this short video on understanding of the justification for dissolution specifications. Now, the dissolution specifications are based on the scientific and regulatory understanding, and the approaches are used from the pharmaceutical development. See the purpose of the dissolution specification. So the specification is a criteria for the dissolution testing to ensure the consistent in vivo performance and the product quality. Dissolution specifications serve as a critical QC tool for batch-to-batch consistency, and dissolution specifications are required to align with the regulatory expectations like ICH, FDA, EMA, WHO. So these regulatory agencies have provided their expectations through the guidelines. And if we follow all the guidelines related to the dissolutions, then it will become very easy to provide the justification for the specification. Key justification parameters are like BCS classification. If you have a product which is immediate release containing BCS class 1 or 3 drug substance, and if it is showing rapid release or very rapid release, that time set the specification like ATQ in 15 minutes or 30 minutes, so that you will be justifying the BCS based biowaivers. So the criteria is that the product should release the drug substance or API more than 85% in 15 minutes or 30 minutes. So for BCS class 1 drugs it will be 30 minutes, and for BCS class 3 drugs it will be 15 minutes. Always use the mild condition for the dissolution testing and follow the guidelines. Then for BCS class 2 and 4, as these APIs are insoluble or low soluble, so these need strong in vitro and in vivo support. That means biobatch dissolution will serve a criteria for setting the dissolution. Clinical bioequivalence data, so align the dissolution profile with BE data or justify through the IVIVC. Method and specification setting, so discriminatory method should be there which can detect the changes in CMAs, CPPs and storage. It can detect the changes in API, excipients or processes. Specification example I have given here, like not less than 80% Q in 15 minutes. So the formulation should release more than 80% in 30 minutes. So S1, S2, S3 stages will be there if the product is IR. Reference product comparison for the generics. So compare the reference formulation, provide the stability data as a support for the justification of specification. Profile remains within the limits during the shelf life. Shelf life data for the dissolution is required to be compiled and provided and used for the justification for the dissolution specification. Best practices I have included here. So base specification on scientific evidence like clinical studies or clinical batches data, BCS classification and the development data. Use validated and discriminatory methods. Justify with comparative and stability data. Ensure method reflects the product performance across the life cycle. So whatever the method you finalize, whatever the specification you finalize, these all should have the scientific evidence and should have a rationale based on the stability studies, based on the development studies, based on the BCS classification, product design like IR, ER, DR and some others, and also should have clinical relevance. So this is the information on specification for the dissolution and its justification. If you have any other points, please comment. If you have any question, you can mail me and you can comment. I will try to answer your questions. Thank you.